Hello, everyone, and welcome to Indoor Biotechnologies webinar on allergen variability and early introduction foods. What have we learned? Where we'll be discussing today's topic of the use of Maria for foods to compare the allergen content of early introduction food brands. We really appreciate you taking time out of your busy schedules to join us to discuss this important topic and are excited at the opportunity to present it to you. A couple of disclaimers before we get started. The products we're going to be discussing today were analyzed by us at InBio from December 2019 through June of 2020. The discussions concern the results of this analysis of the content of certain product samples tested at a certain point in time. InBio does not make any statement about the content of products or product samples it did not test, and it's possible that these products have been modified by the manufacturer since testing. InBio does not advocate for one product over another, nor does it make any product recommendations. We also have no financial interest with the companies listed herein. So we've got a great lineup of speakers for you today, starting with Dr. Martin Chapman, President and CEO of Indoor Biotechnologies. Martin is also a fellow at the American Academy of Allergy, Asthma, and Immunology, and formerly a professor of medicine and microbiology at the University of Virginia. Martin will be going more in depth on the early introduction food study we completed, providing details, data, and the impact of that study. Next, we have Stephanie Phillip, who is our Director of Laboratory Services. Stephanie has been heavily involved in the development and method validation for Maria for Foods. She will be providing more information on that technology. And then finally, we have Zach Schumacher, who is our mass spec specialist here at InBio. Zach will be providing some complementary mass spec data to that early introduction food study. Our moderators today are Dr. Sabina Wunschman, who is VP of Allergen Manufacturing here at InBio, as well as R&D, and myself, Beatty Sturgill, Business Development Manager for InBio. To provide a quick structure of our webinar, we'll first be discussing Maria for Foods with the goal of understanding how Maria for Foods can be used to measure the specific food allergens. Next, we'll move into the early introduction food study, where we'll discuss the details, data, and the impact of that study. The goal of that will be to understand how InBio's molecular approach to food allergy can improve not only the quality of early introduction foods, but other therapeutic products as well. And then we'll move into the Q&A section. I ask that you please submit your questions using the Q&A feature in the Zoom toolbar. You can click on that and type a question. You can post it anonymously or associate your name with it. And we'll get through as many questions as we can at the end of the webinar. One other point, this webinar is also being recorded, which we will send out a link in the coming days of the recording for you for your review or to disseminate as you wish. But with that, I think we're ready to get started, and I'll turn the webinar over to Martin.
Thanks, Beatty. First of all, welcome everyone to Indoor Biotechnologies. We have a diverse group of participants on the webinar. We have allergists and medical professionals. We have representatives of the food industry, early intervention food companies, allergen manufacturing companies, as well as food allergy researchers, people involved in regulation, and food allergy advocacy organizations. So we're really pleased, to have this group available. We also have one or two moms on the call. Because at the end of the day, what we're all trying to do is to produce better approaches towards the treatment and prevention and cure of food allergy. Indoor Biotechnologies has been in this area for over 10 years now. We've adopted what we're calling the molecular approach to food allergy, which is based on 4 principal components. One is the use of purified food allergen proteins to develop better tests and to investigate food allergy. These are the specific proteins that cause allergic reactions in the majority of food allergic patients. coupled with that, we have precise measurement of food, these specific proteins. We can do that by either ELISA. We have a ELISA 2.0, which can measure each individual allergen in about 2 hours. But the problem with that is that if you want to measure 10 different allergens, you don't really want to be setting up 10 different assays. And so to that extent, we've developed Maria for Foods, which is multiplex technology for simultaneous allergen measurements. Now, the acronym Maria stands for Multiplex Array for Indoor Allergens, and Indoor Biotechnologies has developed that over the past 10 years for indoor allergy, for measuring, for example, dust mite cat or dog allergens in houses. The technology has been widely used by the National Institutes of Health and other organizations in, for example, inner city asthma studies, in school studies for assessing environmental exposure to those allergens. And we've been working over the past several years to modify that so we can have multiplex technology for measuring foods. We also have been over the past 2 years, we have been doing mass spectrometry for detecting multiple allergens. This is a total game changer for us. It's enabling us to do different kinds of experiments, to do different kinds of analytics that are not antibody dependent, and it's particularly suitable for food allergens. And finally, really exciting, we're working on IgE monoclonal antibodies from food allergic patients. This is through Dr. Anna Pomeyes, our research director, in a collaboration with Scott Smith at the Vanderbilt University, and we're working on now these patient-derived antibodies, IgE antibodies, to produce better diagnostics and also for more fundamental studies of food allergy. Next slide, please. So, so really what we're talking about, food allergen proteins. If we look at the foods on the right-hand side of this slide, we can see hazelnuts, peanuts, tree nut, cashew, wheat. A lot of the ELISAs that have been current for in the food industry measure what are called total peanut protein. The problem with many of these assays is that we don't know particularly what allergens they measure, and the assays produced by different companies are not necessarily directly comparable. So our approach is to measure the active ingredients, the food allergen proteins that specifically cause allergic reactions. So for peanut, for example, this would be RH1, 2, 3, and 6. Between 50 and 95% of allergic patients make IgE antibody responses to these allergens. The other thing is that we also now know a tremendous amount about the structure and biology of these allergens. Examples of the three-dimensional structures of 4 allergens are listed there. You can see peanut allergen, soybean allergen, GlyM5, egg allergen, GALD1, and BOSD5. So we know what the molecular structures of these proteins are, and they've been extensively studied. So we've posted examples there of all these different allergens. Now, the key element then is, well, it used to be, well, we can't possibly measure all these allergens. That's why we need to measure the total food. But in fact, we now can do that with advances in technology. And so I'm going to hand over now to Stephanie Phillip, who's been developing our Maria for Foods, and she's going to tell you how that works, and then we'll get back to talking about the early introduction foods. So Stephanie, over to you.
Thanks, Martin. So Maria for Foods uses the Luminex XMAP technology in which 6.5 micron magnetic polystyrene beads function as the surface for building 2-site immunoassays. This feature allows a simultaneous measurement of multiple analytes compared to ELISA, where only one analyte can be measured at a time due to the limitation of one binding surface, typically a well of a microplate. The bead sets are distinguishable by unique internal fluorescent dye ratios and are covalently coupled to allergen-specific monoclonal or polyclonal antibodies. So on the right here, there are 3 examples of the 2-site immunoassay that is built onto these bead sets. Each bead set has a different internal dye represented by the different colors here, and they also are ID'd by a number. So for instance, bead ID number 9 is we bound or used for RH1. and bound the monoclonal antibody specific to Rh1 4E2. So immunoassay allergen standards and food extracts are then incubated in solution with the mixture of bead sets, followed by a wash step to remove non-bound material. So in this instance, the Rh1 in solution has now bound to the specific Rh1 monoclonal antibody 4E2. The captured allergens are then incubated with a biotinylated secondary antibody cocktail, followed by a second wash step to remove non-bound material. So here the biotinylated monoclonal antibody 2F2 has now bound to the attached Rh1 allergen. The reactions are then developed by the addition of streptavidin, phycoerythrin, and then read on the Luminex instrument. So beads pass by two lasers in the instrument. A red laser detects the bead's internal dye ratio, which identifies the allergen being measured. And the green laser detects the fluorescence intensity from the streptavidin phycoerythrin bound at the terminus of the two-site immunoassay. Next slide, please. So the first stage in developing Maria for Foods was the purification of natural or recombinant food allergens. Allergens were purified by affinity chromatography and or HPLC, and protein identity and purity were verified by mass spec, SDS page, and amino acid analysis. Allergens are purified and validated via the processes described under an ISO 9001 certified quality management system. And once validated, the allergens were used in the production of monoclonal and polyclonal antibodies and to formulate immunoassay standards. Next. Target food allergens were chosen for assay development based on their clinical relevance, relative abundance, purity, and inclusion in food allergen regulations worldwide. Antibodies were produced and evaluated to determine antibody-antigen affinity, binding kinetics, and cross-reactivity in order to create two-site immunoassays. So in the table to the right, we have each of the 17 food targets, the allergens, and then the capture antibody developed for that assay, and the allergen used in the assay development, whether that be a recombinant or natural, and then the bead sets used for each allergen and detector antibody listed. So all components of Maria for Foods were validated and standardized. Next, please. Food allergen immunoassays standard curves vary according to the allergen being measured over a 5 log range from 0.01 to 1,000 nanograms per mil. Starting concentrations were optimized to produce sensitive assays with a maximum usable range. And full method validations were performed to determine parameters of linearity, range, limits of quantification and detection, accuracy, and precision for each assay. So here on the left is an overlay of all 17 allergen control curves. As you can see, the shape and signals vary for each allergen. To the right are two of the lower MFI intensity control curves. In the curves, you can see that the sigmoidal shape is maintained, and they have maximum linear range, which makes the curve suitable for measuring allergen concentrations. Next. Maria for Foods was then evaluated as a multiplex assay compared to ELISA 2.0 to assess potential compatibility issues once the food allergen bead sets were combined in solution. The allergen concentrations measured simultaneously in 8 food samples using the multiplex array were comparable to ELISA, indicating there are no adverse interactions between these allergens. Next. Finally, as proof of principle, Maria for Foods was used to analyze specific allergen levels in NIST and MonoQA food reference materials. To our knowledge, individual allergen levels had not been measured previously. the food reference materials contained high levels of the relevant allergens and were below detection limit or negligible for unrelated allergens, confirming the multiplex array is highly sensitive and allergen specific. So in the table here, the food reference materials are listed on the left, and allergen concentration results are listed there. And as you can see, for the MonoQA and NIST milk reference materials, the highest concentration of allergen detected was BOSD5, which is a milk allergen. And this was very consistent with the rest of the reference materials, where the egg material was only positive for GALD1 or GALD2, soy GLIAM5, peanut RH3 and 6, and hazelnut core A9. So that concludes my presentation on the development of Maria for Foods. I'll now pass it back to Martin, who will discuss our findings on early introduction foods.
Thanks, Stephanie. Well, let's talk a little bit about the LEAP study. Most of you on the call will be familiar with this study, a seminal study by George Dutoit and Gideon Lack on the control trial of peanut consumption for prevention of peanut allergy. And clearly this created a paradigm shift within the medical community because instead of practicing allergen avoidance, the guidelines were changed so that we now focus, especially for peanut, on trying to have infants consume peanut in early life as a means of prevention of peanut allergy. And one of the main foods that was used in that study was Bamba. Bamba was given to 70% of the children, the infants, in the first year of life. And then progressively over the 5 years of study, they switched to other peanut things, particularly peanut butter and peanut soup. So Bamba was a key component of that study. And in fact, it's the only component so far for which we have validated clinical data of its effectiveness. Now, you can see that by the time the slide was prepared, Virginia were actually the national champions for basketball, which we're all very pleased about. And apparently they're still national champions until this month. So we're even more delighted about that. Next slide. So we measured the allergen content of Bamba a couple of years ago by ELISA. We measured 16 samples from the US and the UK and found that this was a highly well-formulated peanut product with very consistent allergen levels. It was almost like a pharmaceutical grade food material. And by comparison there, we also included Erdnusslocken as a control, really, as a positive control. This is not, it's a peanut puff marketed and used in Germany as a snack for adults. It contains pieces of peanut and therefore is not suitable for children. But you can see also for that product, the allergen levels are lower, but they're nonetheless tightly consistent. And we estimated as a result of this study that there were cumulative weekly doses of about 330 milligrams of peanut allergens in Bamba that were associated with this clinical efficacy. Next slide. Now, since this paper, the LEAP study, it really created an entrepreneurial sort of explosion in the food industry here. A lot of products were developed as early introduction foods, and those are the ones that were included in the study on this slide. We've used a sort of 2 or 3 letter kind of airport code to distinguish these products as we move through this. I should say from the outset that these products were developed, Maria for Foods hadn't been developed when these products came onto the market, and we really aren't very much aware of how these individual products are formulated and compared. Next slide. The protocol really here was we took early introduction foods from 9 manufacturers and 4 controls. We had about 1 to 8 samples of 32 different products that were analyzed for 17 allergens using Maria for Foods. For technical reasons, RH2 was measured by ELISA 2.0. So in total, we had 86 samples tested and 1,541 data points. The foods at the bottom there, PB2, Wow Butter, Erdnusslocken, and Meridian, are really just control foods that we wanted to put in to get an estimate of their levels. And we'll come back to that in a second. Next slide, please. The key findings of this study were, first of all, about half of the products had what we call consistent allergen profiles. That is, they had significant levels of allergen which were comparable to Bamba. And so we kind of were really talking about a sort of Bamba standard for this. And that was the case for 3 of the peanut puff products and 4 out of 7 of the products that were peanut powders. On the other hand, we also noticed differences between allergen levels in different products. Significant variability between allergen levels, especially in multi-allergen products. Because we could measure 17 allergens, it was also a good way to look at cross-contamination. And we found trace amounts of milk and egg allergens in peanut products. We could also find what we called inconsistent allergen profiles in some products. Next slide. This is selected data. The Excel file for this is huge. What we decided to do was initially analyze peanut, milk, and egg products because those are most of the products belonged in that group. If you look halfway down this slide, you can see the allergen levels for Bamba. And basically everything in green are levels that we would regard as being sort of consistent or as in the same ballpark as Bamba. You will note that if you look at lines 2 and 3, we found milk allergen contamination in one of the peanut powders, up to 10 micrograms per gram. Also in the product there, which was milk powder, which we believe is produced in the same facility that also produces milk and egg, we could detect very trace amounts of milk and egg in that product, of egg and peanut in that product too. On the other hand, if we looked at another group of product, mixed food powder and other fruit sources, we found significantly lower allergen levels. We found inconsistencies in the ratios of different allergens. For example, if you look at the bottom 2 products there, you can see that they contain low levels but detectable levels of RH2 and RH6, but the levels of RH1 and RH3 are almost completely gone, and the reasons for that are unclear. Next slide. These were the control foods. Part of the reason for doing this is PB2 is used by quite a few allergists to actually formulate products for their own oral immunotherapy and other treatments. Peanut butter is also recommended for treatment to give to infants as a potential alternative to Bamba. And we use soy butter there as a control, and we didn't detect any allergens in it. Next slide. These are the peanut allergen levels in food puffs. We analyzed 5 samples of each puff product. You can see the levels there for Bamba. In all of the 3 cases on the left, the highest levels were for RH3. So we got Bamba, Erdnusslocken, and another peanut puff. By contrast, we were unable to detect these allergens in a multi-allergen blend food puff on the right. Next slide. Now, two EIF products involve giving doses of allergen over different days. This is one such product, and you can see it starts out with at a lowish level, around 100 to 1,000 micrograms per gram day 1. Then progressively over the course of 7 or 8 days, this rises to about 10-fold for each of the 4 allergens, and is consistent. Now, in this particular product, we did detect trace contamination with milk at levels ranging from 0.6 to 10 micrograms per gram. Next slide. Another product involved giving milk on day 1 to day 4, milk and egg day 5 to day 8, milk, egg, and peanut 9 to 11, and finally a higher dose of milk, egg, and peanut at day 12 to 15. And you can see that we can monitor that significant levels of milk and egg and peanut allergens in this particular formulation. What we did notice was that over the course of the days, as these products are mixed together, you can see that the milk allergens in blue decrease over time. And we think that's actually what happens because this is being mixed. So you take some milk, you take some egg, and you take some peanut, you mix it, and as that mixing goes on, the levels become lower. But nonetheless, significant allergen levels in these particular products, and consistent with the Bamba analysis. Next slide. By contrast, when we look at Multi-blend, early introduction foods, and fruit sources, we found generally lower levels of these allergens. We've included a line there that I mentioned previously, trace contamination at 10 micrograms, which is that dotted line there. This is an arbitrary value, but at the same time, you can see quite a few of these products were below that level. And the levels varied, you know, for some of the manufacturers, for example, the reason we did this was because you have a manufacturer that produces a mango and soy sauce, or peanut and apple, or pear and egg. So these are all individual products that we tested with this multi-allergen screen. And then the one product line there, which is multi-allergen blend of 14 different allergens. You know, for milk, for example, we detected high levels of milk in that product, but then for many of the other allergens, the levels were generally lower than some of the other EIF that we tested. Next slide. So what are the study limitations, and what are the potential causes of variability in these early introduction foods? Well, you know, during the review process for our paper, which is has recently been accepted for publication, some of the reviewers raised concerns about extraction conditions. Could this affect what was going on? We don't think this is the case, because Stephanie and her group tested 3 different extraction buffers and conditions before we started this work, and we did try to optimize this for the allergens that we looked at. The other limitation really is that for quite a few of the allergens, anything other than peanut, milk, and egg, we're measuring a single major allergen, and we're assuming that that measurement correlates with other allergens present in the food. Further work would need to be done to confirm that. Obviously, we're dependent on the antibodies that we use in the test, and it's possible that if some epitopes are denatured as a result of food processing, we might, you know, underestimate the results. So on the other hand, if we had a food that didn't contain the major allergens that we're measuring, and bearing in mind that the major allergen is one to which more than 50% of allergic patients respond. So if we could not detect that allergen, that would be a cause for concern. As far as the early introduction foods are concerned, what could cause the variability? Obviously, the quality and the way in which the food source ingredients are prepared and validated, how they're formulated, mixed, or blended as part of the processes. Food processing itself could lead to thermal denaturation or acid precipitation, potentially, of potential allergens. There could be aggregate formation, which could contribute to the variability, and other potential denaturant effects. We don't know how these foods are actually prepared, and so unless we knew more about the process, we couldn't really comment any further on that. Next slide. FDA, one of the EIF companies applied to the FDA for a qualified health claim. This claim, and that was exhaustively examined by the FDA, by Dr. Ballantine, and the link to that is given below. This was a 40-page analysis and response to this request. And what you see here in terms of what the FDA is saying is that that it's giving this claim on the basis that there is evidence that for peanut, one can reduce the risk of developing a peanut allergy. But the FDA determined that the evidence supporting this claim is limited to one study, which is a LEAP study. There have been subsequent studies which were not necessarily considered in this analysis. And then the important caveat that if your infant has severe eczema or egg allergy, check with your infant's healthcare provider before feeding these foods containing ground peanuts. I have to say that Dr. Ballantine's, it was a clear, objective evaluation of the evidence, under which the FDA operated to provide this health claim. So far as we're aware, there are no similar claims that have been provided for example, milk or egg. Next slide. So as far as the food industry is concerned, we now have a Maria for Food 17-plex that covers all of the regulated allergens, both in the US and in Europe. So we think that this offers several advantages compared to other methods for allergen measurement, and is potentially something that can be used in the food industry to control and measure allergen levels or in different foods. Next slide. So Maria for foods then. We can measure multiple allergens with sensitivity, accuracy, and precision. I really want to stress that all the components in Maria for foods are standardized. We can define the antibodies, we can define the reference materials. It also can be used in tandem with mass spectrometry, because basically if you're using mass spectrometry for foods, you're also measuring specific, in most of the cases, you're using peptides from allergens as the marker in the mass spec study. In terms of its applications, screening of allergen contaminations, validation of allergen control processes. And what we want to come out of this study is we can help manufacturers improve their products by measuring specific allergen levels, and whether that applies to early introduction foods, to oral immunotherapy products, or oral food challenge materials. We've actually published some data on that recently. Working with Michelle Makiag and Wanda Botanical at Harvard University, we've measured food allergens in a large school study in the northeastern US, and that is currently being considered for publication. And the technology can also be applied to establishing risk thresholds that are based on actual allergen exposures. So I think at that point, I'm done, and I'm going to pass it over to Beatty and Zach, because we have some new data, exciting new data on mass spec and SDS page analysis of these early introduction food products.
Yes, so thank you, Martin. I would like to share the data that I've collected on these early introduction food products spec, as well as show you what they look like on a more traditional SDS analysis. So to start with, I want to describe our spectrometry, our mass spectrometer, and our data collection conditions. We're using a Q-Exactive paired with a Vanquish LC, and we're preparing these food samples with a traditional trypsin digestion with reduction to allow for a optimized amount of protease activity in these samples. The data collection is using a full MS2 mass or data acquisition method. This allows a broad section of analytes and not focus in on any particular area. We want to see as much as we possibly can with this measurement. It has not been tailored to any particular allergen, which is a possibility for later study. And the data collected from these mass spectrometer data acquisitions was Proteome Discover 2.2 using the Sequest node, and these analysis were accessed on UniProt using the publicly available proteome data specific species and their food groups. So if I can move on to the next slide. I have some Maria and mass spec data that I want to show you how. So this table here, we have 4 products on the left, and above we have 3 food groups and their relevant allergens for the Maria analysis. Our Maria data here is displayed in micrograms per grams. Our mass spec data is a combined mass spec signal intensity of the relevant peanut, milk, and egg allergens, respectively, shown in parentheses cells. Each of these samples had effectively, this analysis could be performed on each of these samples with only 1 data collection, with 3 things for the peanut, milk, and eggs. So that's how we've produced this data. And you'll notice that when we get spikes in our levels for a peanut, milk, or egg allergen, we see a corresponding spike intensity, a signal intensity as well. There is an exception. The SFO food powder, BOSD 11, was notably high by Maria, and a corresponding increase in the mass spec signal intensity that mapped back the peptides from BOSD11 as well. And the samples indicated in pink are regarded a low intensity response by mass spec. These are typically characterized by peptide signal level, the E to the 6 range. For these samples, we saw peptide response anywhere E to the 5 to E to the 10. So that gives you an idea of the broad range intensity that we're working with here. And while this thermometer allows us to look at specific allergens, specific proteins in a sample, we can also take a step back broadly. So if I could move to the next slide, I can show you a broad look at what all of these have in them. So we have on the left 16 different EIF products, excuse me, that we've looked at, and on top we have different food sources. Of these 19 food sources, we've detected allergens from 17 across these 16 food products. Any positive indication, color and a plus is representative of an analysis that yielded 2 allergens per food source, 2 confident identifications of allergens per food source, with the exception of food such as cod and oat that don't have such a robust representation of allergens in their proteomically available data. And that also brings up the data used to reference against for the proteome samples is important. If there isn't enough robust and high quality data, the quality of your analysis will suffer. So that factor is something that's important, as there is not an equal level of data available for all of these food sources. We're going to continue to look at in an ongoing basis in the future. One addition can be used to look at from all 19 of these different food sources. So it's a very powerful way to look at a broad section of analytes. And now that I've shown you this mass spec data, I'd like to just have a look at the age data. You'll see on the left, you have a set of puff products that have a more traditional and expected peanut allergen banding pattern, red, you'll note. On the right gel, we have the food powder product that contain peanut as well as some other food sources, particularly the RSS in the center of the second gel contains both egg and milk as well. So you'll notice in these products, some of them have the expected that is exactly what we would expect to see for a typical sample representing peanut allergen. Some of them do not have these banding patterns, and go back to our Maria data, our mass spec data, and put together a comprehensive of what is actually in these samples, what do we think is happening, and how we can address any potential limitations with analysis methods. So this is a great look at these products and produce a high composite result. So with that, I can this back over to Beatty. Thank you very much.
Thank you, Zach, and thanks, Martin and Stephanie, for your all's presentation as well. Before we move into Q&A, this would be a good time to just kind of tidy everything up and again hit on what Martin mentioned earlier as our molecular approach to food allergy. Here you can see all the resources that InBio has to offer, starting with the Maria for Foods. We've discussed that at length today. We offer this as a kit or a service here in our lab. The ELISA 2.0, which are pre-coated plates with all the reagents you need to complete the assay in 2 hours. We also have a comprehensive portfolio of purified food allergens that are manufactured under an ISO 9001 QMS system. Martin mentioned our human IgE monoclonal antibodies. That is an exclusive product in Bio that we're very excited about. We have those for food allergens as well. Zach mentioned our mass spec suite. We have several standard services available to our clients, ranging from protein identification to intact mass in our mass spec capabilities. And then, of course, we also offer contract research opportunities as well for food, the food industry, food allergens. What that could look like would be custom protein purification, immunoassay development, custom allergen extractions, or characterizations of food products and food immunotherapies. We have 2 papers relating to Maria for Foods and the early intro to foods study. The first one being doses of specific allergens in early introduction foods for prevention of food allergy. This has been submitted and accepted by the Journal of Allergy Clinical Immunology in Practice. And over the next several days, we expect that to be an article in press, and we'll be following up with all attendees with the link to that paper. We've also submitted a paper, the simultaneous quantification of major food allergens using fluorescent multiplex array. So another thing we're really excited about here at Indoor is a recently signed distribution agreement with MilliporeSigma. They will become a global distributor of our purified food allergens. And again, that's a comprehensive range of both natural and recombinant food allergen analytical standards. The transition of these 50 or so products is expected to be completed in the coming months. We're really excited about this opportunity and think it's a testament to the importance of high quality products to be available for food allergy research, diagnostics, and immunotherapies. So with that, we can move into the Q&A. I see we've got a good bit of questions submitted here. Again, just to reiterate, I ask that you please submit your questions using the Q&A feature in the toolbar. If what we can't get to today, we'll follow up. If you leave your name with the question asked, we'll be sure to follow up with that as well. And I'll stop my screen share here. And Sabine, I don't know if you have some questions to start us off.
Yeah, I do have some collected here, so we can start at the beginning. About the Maria analysis and ELISA, the first question was, can you share any data comparing results of an ELISA and Maria analysis? And does the sum of RH1, 2, and 6 in a sample equal the ELISA analysis of total peanut?
Stephanie, you're muted.
Thank you. The total peanut assays that are commercially available are usually typically measuring 1 or 2 different allergens. So we don't really have a good comparison of our specific allergen ELISAs and Maria versus the total peanut assays that are available.
Mm-hmm. Oh, well, I think I can add that from the mass spec analysis, it seems if we look at the relative abundance of the different peanut allergens, RH1, 2, 3, and 6, and 7, pretty much would represent the majority of the peanut proteins available that are present in a peanut flour sample.
Yeah, I think it's a good point. Yeah, I think it's a good point. You know, there was a study done by Shmali Jayasena 5 years ago, which showed that 5 out of the 6 peanut assays that are commercially available measure predominantly RH3, and some measure a bit of RH1. Another assay measured primarily RH2 and RH6, and that's why we really need assays to measure all of the allergens, and that's what we've been trying to do here. So we think that if we can measure RH1, 2, 3, and 6, then we're pretty well measuring the allergens that we need to measure.
The next question is, I see wheat but not gluten. Can gluten be included in a Maria test?
So yes, so the first stage in doing an assay development to add to Maria is actually having the analyte for antibody production. So our group relies heavily on those available allergens from our allergen group, which Sabina heads. So I don't know if there's plans for a gluten, but she might be able to comment a little bit more on that.
Yeah, so essentially the Tri-19 is what we're using in the Maria, correct, Stephanie? So that is an omega-5 gliadin, and the gliadin is part of the of gluten, but there are several other allergens that are gliadins and glutenins. So we are only really having one allergen at the moment, but we are looking into purifying other gluten allergens as well. So the next question is from Svetlana Starchenko, commenting on some of the food allergens are recombinant. How similar are the structure and post-translational properties of the recombinant allergens in comparison with the native? So I guess I'll take that one. So we don't necessarily produce, if we express a recombinant allergen, we design it to be expressed either on E. coli or in Pichia, pretty much orienting ourselves on what has been published for this allergen. And we don't necessarily also purify the natural counterpart of it. But what I can say is that the allergens are tested for immunoreactivity, and in this case they're obviously used in the Maria for food, so they have good immunoreactivity with monoclonal antibodies, and every allergen is also tested for IgE reactivity before we, as part of our regular validation. So the allergens are correctly folded to the extent that they're immunoreactive and can be used for diagnostic assays. They're IgE and IgG reactive allergens. They have the similar molecular weight, and if there's excessive post-translational modifications expected, such as may be the case in Pichia, we would go ahead and silence N-glycosylation sites. I guess that's probably answering the question. For some allergens that we have a natural and recombinant counterpart, we have done comparisons, and they usually correlate very well for IgG binding and IgE binding. The next question is also from Svetlana Starchenko. It's commenting on the Maria platform. Is it applicable for measuring allergen levels in diagnostic and therapeutic products for subcutaneous vaccines for allergy treatment?
I would say that the Maria is applicable for most any situation where you would want to measure specific allergens. We're currently involved in several studies right now that are using the technology. So I would say yes.
Okay.
I guess I would probably put one qualifier in there, and I think it depends on what allergens you're doing. You know, the thing about Maria is that it's exquisitely sensitive, and for some of the milk products we were having to use, Stephanie was having to dilute these out to 1 in a million. So it depends. If you're going to, for example, just measure a couple of allergens and you don't want to dilute out excessively, maybe you might want to use ELISA for those, you know, the ELISA 2.0 assays that we have. I think a lot of it depends a bit on the application, and whether or not you're using a natural allergen extract or a modified food extract. I think all those need to be looked at. Yeah.
The next question is from Rick Goodman. There are some food sources that are complex, such as fish, carbon, and the proteins are not identical across the species. Would you recommend more broad testing for some of these before using it for general food evaluations?
So we're still currently researching different fish extracts as we're able to get them in our hands, just to see and be certain what exactly we can measure with our CypC1 assay. So broad testing, I think, is quite important for these food evaluations.
Okay. Christina Capule, do you have plans to add additional allergens to your Maria for food kits?
So yes, we're always looking to add new allergens to the Plex. Right now, we're probably going to focus more on adding some additional tree nut allergens in the future.
Yeah, and if I may add to that, so currently we're working on a couple of Brazil nut allergens, beret 1 and 2, on pecan allergens, pine nut allergens, and also we have recently released almond allergens. And I think we are going to add pistachio allergen as well as kiwi and strawberry. So I think there will be many more food allergens being purified, and then we decide which ones we would like to raise antibodies against and add them to the Maria for foods. The next one is also from Rick Goodman. How do you choose which isoforms to use for some of the proteins? They can be quite different, and maybe in natural foods as well. So for the recombinant allergens, that is an issue. We usually orient ourselves again on what has been published in the literature. Are there different isoforms that have been already expressed, and are there differences in terms of IgE reactivity? If there's not much to be found, we usually just decide for one. The other thing we oftentimes do now that we have a mass spec is we look at the predominant isoform that is present in natural material and then go from there. There's an anonymous question that is about what is the accepted minimum microgram per gram that trigger a reaction? Martin?
I'm actually reading the question. It's quite a long question. I think that a lot of these issues about micrograms per gram in terms of peanut either consumption or involvement in allergic reactions, especially as part of foods, are still things that we need to work out. The comment also addresses the EAT study. And we referenced that in the paper, particularly because it showed some effects of egg as a preventative for food allergy. But I think a lot of the dosing issues need to be worked out. Part of the problem also is that you can't really do these studies with purified peanut proteins unless they're produced under GMP conditions, and so far that's actually not usually the case. So, I mean, obviously you can use foods where you know the concentration of the allergen in the food. One of the interesting things that a study that we published last year in terms of this was looking at oral food challenge products, and we actually looked at milk muffins. this was based on a study you know, 3 years ago, there was a tragic case of a child that had anaphylaxis and sadly died as a result of consumption of muffins, and in a double-blind placebo-controlled food challenge. As a result of that, we went ahead and did an analysis, because we had these assays for BOSD5 and BOSD11, of the allergen content when you bake muffins. What happens? And what happens is that when you do that, the BOSD11 is completely BOSD5, let me get it right, is completely destroyed, but the BOSD11 is retained, and about 70 to 80% of its activity is retained after baking with IgE antibodies. And so what we would expect is that there may be some issues with food challenges with these kinds of materials, especially in patients who are strongly allergic to BOSD11 and have IgE levels of greater than 20 kilounits per liter. So I think that I mean, it's a very good question. We don't have all the answers. And then the other aspect to it is, how do we compare the specific allergen measurements with measurements of peanut protein and how those are done in the food industry? And I think those are all things that we're really very interested to look at.
Thank you, Martin.
Sabina, if I may interrupt just briefly.
Yes.
This is probably a good time to put up a poll to see. I'll post it now. You know, at InBio, we're committed to helping the food industry and the early intro foods manufacturers work through these issues, and I would be happy to host a roundtable discussion in the future. So I put up a poll. If you would, you know, if you answer yes, we can reach out in the future to schedule maybe a more interactive webinar where we can open up for, you know, shared video discussions and things like that. So I'll just have that running in the background.
So if you run that in the background, maybe we can do one final question from Chang Cho. What is your approach in correlating the detection amount to the total protein in natural product?
I guess I'm not quite sure of the. I mean, we haven't looked at total protein primarily because we don't trust most total protein assays. I mean, you know, the assays that are out there have been running for 50 years. That, you know, it's very. It gives you an answer, but whether that is meaningful, particularly meaningful, I don't think is clear. And it's certainly something that I think warrants further discussion. I think it's a great question, but we haven't done that yet. We haven't actually related the total protein in these materials to the specific allergen levels. We may have done some work at one point years ago with peanut, but we haven't really pursued it with other allergens.
All right.
Well, that takes us to 12 o'clock here. So we've reached the end of our webinar, but do appreciate all the questions and attendees, again, taking their time out of the day. Appreciate all the speakers here at InBio for putting together the great presentations. I'll be following up via email with all attendees with links to a recording of the webinar, some other helpful resources, the papers, etc. And we can expect that probably by the end of the week, early next week. But other than that, speakers, do you all have anything else you'd like to share before we close here?
I would just like to say we've enjoyed making these presentations. We've enjoyed listening to the comments. We're dedicated to working towards creating improvements in these products and other products for food allergy treatment. And so anything that we haven't covered, anytime you want to get in touch, please get in touch with Beatty or Sabina or me, Stephanie, and let us know how we can help. Thanks very much.
Yeah. All right. Thanks, everyone. And we'll speak soon.
Thank you.
All right.
Thanks a lot.
Thank
Bye.
you.
Bye.